Okay, folks, let's get started. A lot of questions of what was on the screen, so let me just say them for everybody. I thought I had it labeled, but I didn't, so no wonder there's confusion. It says 200 points. You didn't have 200 points, and no, you can't add 100 to your score to get what you have. The 200 points are the sum of your exam 1 and exam 2. So if you made a 50 on the first exam and a 90 on the second exam, you have 140. There's about where you stand. Okay? And people say, well, do you give pluses and minuses? And absolutely I do. All right? So, there's rare, and the A minuses, there's the B pluses, B minuses, C, anyway. So you see basically where you are in the class with this. So add up your 2 exam scores and plot it on there, and you will know. I'll make a note on the schedule page later indicating that it is, in fact, the sum of those 2. Okay? So as I said last night, on the email I sent out, the average on the exam was 70.6. The low was 12. The high was 100. So obviously performance was better, and I was quite happy with that. In general, people went up. There's always some people who go down, but in general there was an upward trend on people's scores, and I was, of course, happy to see that. So that tells me that you're learning more. And this was be honest with you, this was a difficult set of material. A lot of material here, and when I see the exam average go up like that, then that obviously makes me feel better about how you're doing in the class. So happy to see that. I've also had several questions on the things I talked about last time with respect to why bypassing that step in fructose metabolism causes you to become fat. So I'll go ahead and say a little bit about that right now. So if we look at this, which I talked about last time, I said that what this is doing is it's bypassing the phosphofructokinase step of glycolysis. And I probably just didn't fill in the blanks after that, and I apologize if that was the case. So why does bypassing the phosphofructokinase make you fat? Well, if you bypass phosphofructokinase, then the rest of reactions of glycolysis will continue. And that means a couple of things. One, you're going to make more ATP, which by itself isn't bad, but we'll see why that is in a second. And second, you're making more pyruvate, which is the end product of glycolysis. So if you're bypassing that step, you're force-feeding the cells to make ATP and to make pyruvate. You're also making more NADH. Okay? Now, pyruvate is readily made into acetyl-CoA. We'll see that next term. And acetyl-CoA is a precursor of fatty acids. So now you've got a precursor of fatty acids, you've got energy in the form of ATP, and you've got a reductant in the form of NADH. You're primed to make fat. So when you take high fructose corn syrup, in my opinion, you're actually favoring the production of fatty acids and fats considerably. The more you have, the more likely you are to produce those. Okay, that makes sense? Clear as mud? Pretty good turnout for a day before Thanksgiving. I'm happy to see you guys here, so this is good. Okay, let's see. So what I want to do today is I'm going to excuse me, talk about the synthesis of glucose, which is a phenomenon known as gluconeogenesis, and that's kind of a fairly straightforward pathway after you've seen glycolysis. I want to talk a little bit more about regulation, because now when we talk about regulation, we talk about regulation not only of glycolysis, but gluconeogenesis. And we're going to see they exhibit this reciprocal regulation very, very nicely. And last, I'm going to talk about the Cori cycle. So that's what's on tap for today. I start out with a sort of an odd observation, and your book puts it here for a reason, and that is glycerol. Glycerol can be converted into dihydroxyacetone phosphate. And dihydroxyacetone phosphate is an intermediate in glycolysis. So here's entry of yet another thing into the glycolysis pathway. Why is this relevant? This is relevant because when you break down fat, you produce fatty acids and glycerol. And the only component of those that enters glycolysis or gluconeogenesis is the glycerol. So you can either take that glycerol and break it down and get more fatty acids, get more energy. You can take that glycerol, convert it into DHAP, and break it down in glycolysis so that you're making more energy. Alternatively, as we will see, you can take this and you can make it back into glycerol. Make it back into glucose. My mind is not working today. Make it back into glucose. And the reason for mentioning that here is, as we'll talk next term about fatty acid metabolism, this is the only part of a fat that can be made into glycerol. We cannot convert fatty acids into glycerol, but we can convert glycerol into. We cannot convert fatty acids into glucose, all right? But we can convert glycerol into glucose. Now that I have you totally confused. Okay. Does everybody understand what I just confused everybody about? Yes, Laurie.
This reaction could go either way. It can end up producing.
Yeah. So once you get to DHAP, it can make glucose. It can break down. It can break down and go to make pyruvate. Okay. I'm going to switch this battery again. Okay. Hopefully that'll improve things a bit. All right. So that's what happens with glycerol. Excuse me. Let's turn our attention then to the synthesis of glucose. In the synthesis of glucose, you're going to see overlaps considerably with glycolysis. Not absolutely. There are places where there are different reactions, and I'm going to show you those different reactions. But you can see on this schematic, all of the blue enzymes that you see on here are the same enzyme of glycolysis and gluconeogenesis, the only difference being the direction of the reaction. So the only difference between these for the blue enzymes is the direction of the reaction. If we were doing glycolysis, we would start at the top and we would go from glucose all the way down to pyruvate. When we are making glucose in the process of gluconeogenesis, we are starting at pyruvate and working our way upwards. And as we shall see, there are some energy considerations to getting around some of the energy or some of the molecules that we need to make. And there are some barriers that we have to go through. One of the barriers starts at the very first reaction of gluconeogenesis. If you recall, in the last reaction of glycolysis, pyruvate kinase catalyzes the conversion of PEP into pyruvate. And I said that was the Big Bang. That was a very large energy consideration. And as a consequence, it's a big obstacle to overcome. So the cell doesn't try to climb that mountain. It actually goes around the problem by doing two different reactions. So for you trivia hunters out there, there are 7 enzymes of glycolysis that are identical with gluconeogenesis. 3 enzymes of glycolysis are replaced by 4 enzymes of gluconeogenesis. I just throw that out for you just so that you know. Okay? 2 enzymes at the bottom are necessary to get around the pyruvate kinase reaction. Okay? So the first of these is an enzyme called pyruvate carboxylase. And pyruvate carboxylase is an enzyme that's found in the mitochondrion. Now, all the reactions of glycolysis occur in the cytoplasm. We will see that the very first reaction of gluconeogenesis occurs in the mitochondrion. All the other reactions occur in the cytoplasm except for the very last one, and I'll tell you where that occurs when I get to that point. Okay? So the first reaction and the last reaction of gluconeogenesis are the only ones that don't occur in the cytoplasm. So pyruvate carboxylase catalyzes the first one. I'll show you the reaction in a minute. And all the others are occurring in the cytoplasm. You bear with me? Now, looking at this briefly schematically, we see, first of all, a new molecule that's appeared that we didn't see in glycolysis. And this new molecule that's appeared is oxaloacetate. Oxaloacetate is a 4-carbon molecule. It means we had to gain a carbon, and I'll show you that in a second. We also see that there's some significant energy that's necessary to get back to PEP. So we have 2 reactions, and each reaction takes a triphosphate. The first one takes ATP. The second one takes GTP. I also want to remind you that to make glucose, pyruvate is a 3-carbon molecule. So we have to start with 2 pyruvates to get all the way up here to 1 glucose. That means that in making glucose, we have to have 2 ATPs for the 2 pyruvates to get here, and we have to have 2 GTPs to get the 2 oxaloacetates back to PEP. And again, I'll show you these reactions in a second. This schematic also shows some ways that other molecules can enter the process. So here's glycerol I showed you before. Glycerol can come in, it can go up and make glucose. It can go down and make pyruvate. If we look at lactate, lactate, you recall, was the product of fermentation. And lactate is what I describe as a biological dead end. It doesn't go into something else. It can only go back to pyruvate. It can only go back to pyruvate. A good exam question might be, under what conditions will lactate go back to pyruvate? What's it going to take? Based on what we've talked about, what do you suppose it would take? Oxygen. So, after you've had a chance to catch your breath and you stop that heavy exercise that you're doing and your muscles are starting to catch up, they're going to start converting lactate back to pyruvate. And we'll also see that this reaction occurs in the liver in a process I'll show you later today. Okay? But in order to go from lactate back to pyruvate, we have to have oxygen. Because to have oxygen, it takes oxygen for us to have NAD so that we can go back here and make pyruvate. All right. The other thing is that we see here that some amino acids can be brought into the pathway, and they come in through oxaloacetate. A very simple one of these is aspartic acid. If you replace the amine of aspartic acid with an oxygen, you have oxaloacetate. So they're very closely related compounds. And in this way, we see that proteins can serve as precursors of glucose. So for those of you like me who think that you should have low-carb diets, okay, if you eat just fat, you can't make glucose, from what I've been telling you. But you can eat protein and make glucose. So your body needs glucose, and as long as you have a protein source in there, you're going to make glucose. If you don't eat anything, your body will start breaking down your protein so that it can have amino acids to make glucose. So both of these are possible. Okay, well, let's look at the individual reactions. I'll talk about these other reactions up here in just a few minutes. Okay, so pyruvate carboxylase. Pyruvate carboxylase, I mentioned it actually earlier in the term. And pyruvate carboxylase is an enzyme that uses biotin, it's B-I-O-T-I-N, biotin as a coenzyme. And I mentioned at the time that biotin is a coenzyme that we almost always find associated with carrying carbon dioxides so they can be put onto something. So look at what's happening in this reaction. We have pyruvate here. Here's a carbon dioxide, ATP, and we're adding this carbon dioxide on here as a carboxyl group. The upshot of which is we have ADP produced and protons. Okay? So oxaloacetate is right here. This enzyme, pyruvate carboxylase, as I noted, is found only in the mitochondrion. That means that if we're going to start making glucose, we have to start in the mitochondrion, which means we have to put pyruvate into the mitochondrion in order for gluconeogenesis to get started. If we can't put pyruvate into the mitochondrion, we can't make glucose from pyruvate. Well, it turns out that pyruvate normally is dumped in the mitochondrion anyway after gluconeogenesis. And the reason is that's the place where acetyl-CoA is made from pyruvate. So there's plenty of pyruvate floating around in the mitochondrion. That's not a problem. And there's also pyruvate carboxylase floating around in the mitochondrion as well. Okay. And don't need to talk about that. Okay. Here's biotin within the pyruvate carboxylase enzyme. And you can see biotin is linked through a lysine residue. There's biotin. It's actually covalently attached to lysine. And here's the portion down here that grabs onto CO2. Okay? So biotin has literally a handle for holding on to CO2 so that the reaction can proceed. There's the activated CO2. And that's about what I want to say there. Okay. The second enzyme that was necessary to get past that pyruvase kinase reaction of glycolysis is one that's labeled as phosphoenolpyruvate carboxylase, or as you're more likely to call it, PEPCK. PEPCK. Just don't spell it P-E-P-S-I-K. Okay? By the way, this reminds me of something that, and I'll mention it in class next week, is I did a lot of grading on this exam. And as I was grading the exam, first I noticed a lot of people who lost points because they used abbreviations that didn't match the correct ones, so that was unfortunate. But I saw, and I allowed it on this exam, but I'm not going to allow it on the next exam. People don't know how to spell protein. Okay? Now, I think if there's one word that everybody in this class should know how to spell besides their own name, it's protein. So the rule in the final exam is going to be every time we see a misspelled protein, it's going to be a -1 point. So my advice to you would be to make sure you know how to spell protein on the final exam. Okay? Biochem people should know how to spell protein. It's shocking how many people don't know how to spell protein. I'm not going to spell it for you here. I'll let you look it up. Make sure you get that way you'll remember it. That way you'll get it right. Okay. All right. PEPCK. So if you want to use the abbreviation, the proper one is PEPCK, or you can write out phosphoenolpyruvate carboxykinase. This reaction is basically going to take off that carbon dioxide that was put on, and it's going to create a double bond. Okay? And that's necessary to make phosphoenolpyruvate. So we see loss of the carbon dioxide. Here's the carbon dioxide coming off. Here's the phosphate coming on from GTP. And notice the GTP is the energy source. And the upshot of that is phosphoenolpyruvate. Now, if you think back to when I talked about the pyruvate kinase reaction, you may remember I called it the Big Bang because I said there was almost enough energy produced to make 2 triphosphates. These 2 triphosphates that's necessary to bypass that reaction convince us of that. It takes that much energy to get back past that reaction of pyruvate kinase. Okay, well now we're at phosphoenolpyruvate, and we can look back at our overview and say, well, if I'm at phosphoenolpyruvate, I use the same enzyme for glycolysis, so I'm going to run the reaction upwards, just running the glycolysis reaction backwards. I run this reaction upwards, I run this reaction upwards, I run this reaction upwards, and I get up to here. So all these reactions that I see here are just going to go backwards from glycolysis. What do you suppose is going to determine if we're making glucose or we're breaking down glucose? That is, if we're doing glycolysis or gluconeogenesis. What's going to be necessary? What's going to be a big consideration? Yes, sir?
Regulation of the very first enzymes.
So regulation is important, and we'll talk about regulation. But if we have the enzymes, let's say if there weren't that, there's other things to consider. What would you say? Concentrations. Okay? So regulation is going to affect the concentrations of these. So in order to make glucose, I'm going to have more of this, and then make more of this, and then more of this. Because what I'm going to be doing is I'm going to be just pushing upwards. That's what I'm doing in gluconeogenesis. Okay? All right. So these reactions are all occurring in the cytoplasm. Notice that none of these enzymes here are regulated. They're not regulated enzymes. The regulated enzymes down here, the regulated enzymes up here, and up here. There's no regulated enzymes in here. So the forwards or backwards is going to be determined solely by the concentration of reactant and product. The reactant in this case is the thing on the bottom, and the product is, and in this case, is the thing on the top. Whenever we look at delta G zero prime values, we take the negative of the reverse reaction. Okay? So if I have, let's say, the aldolase reaction, which is right here, going down in the glycolysis direction, it's about a +20. Going in the gluconeogenesis direction, it's going to be -20. Okay? So now I don't have an energy barrier to get over in order to do the aldolase reaction. I do have one consideration with respect to oxidation and reduction. What is that consideration? I have to have NADH. Okay? So NADH is necessary here. It's the opposite situation from fermentation, because in this case I want to have NADH, And I'm going to have NADH when the cell is in a pretty good energy state. It's in a pretty good energy state. It's done all its burning it's going to do. It's done all it's made all the ATP it's going to do. And another thing we're going to need here is we're going to need ATP, or triphosphates, because we need triphosphates down here to get it going. Okay? We we don't produce triphosphate here. Actually, we need a triphosphate here in reversing this reaction. So we need a total of 2 ATPs here. We need a total of 2 GTPs here. And we're going to need 2 more ATPs here to make this overall process go. It's going to cost us 6 ATPs to make glucose. It's also going to cost us 2 NADHs to make glucose. Okay? So when the cell has lots of energy, it's going to be making glucose. Okay? All right. Well, this brings us to our next enzyme, which, if we were going in the glycolysis direction, would be PFK, phosphofructokinase. Notice that gluconeogenesis has a different enzyme. The different enzyme it has is called fructose 1,6-bisphosphatase. That's B-I-S-P-H-O-A-T-A-S-E, bisphosphatase. You'll probably call it F16BPAs. F16BPAs. Don't confuse F16BPAs with F16BP. The ASE on the end always tells you it's an enzyme. So F16BPAs is an enzyme. F16BP is a molecule. In fact, it's the molecule that F16BPAs works on. Now, I'd like you to look at the reaction that's going on with F16BPAs. What we see is that F16BPAs is converting F16BP into fructose 6-phosphate. Isn't that just the reverse of the glycolysis reaction? I see several people saying yes. It's not. Okay, I'll give you a hint. There's something different. What's different? What's that? Okay, so the glycolysis direction, we had to use ATP to make this. If we were to try to reverse the glycolysis reaction, we would have to be producing ATP in the reverse direction, right? We're not doing that. We're just simply clipping off a phosphate. Now, there's a couple reasons why that's important. The primary reasons are that it saves the cell energy. Okay? If we run the glycolysis reaction backwards with PFK, we're going to be making ATP. This could be a problem. Why could this be a problem? Well, remember I said when we're going through gluconeogenesis, what does the cell have? It has lots of ATP, right? If the cell has lots of ATP and we try to run that reaction backwards, we're going to have a lot of product to start with. The reaction's not going to be very favorable. So this helps to make the reaction favorable. A second consideration is that the cell already has lots of ATP. It doesn't have much ADP. Do we want to be adding more ATP in the process of this and compounding the problem? No, we don't. So the simple solution is to simply clip that phosphate off. That saves energy, okay, and accomplishes what the cell is after, which is making fructose 6-phosphate. All right, fructose 6-phosphate is converted back to glucose 6-phosphate by phosphoglucoisomerase, just in the reverse direction of glycolysis. But we get up to here and we have exactly the same situation that we had with the PFK reaction. We don't want to run the hexokinase reaction backwards, because if we did, we would have to be making ATP again. Same problem as before. There's no reason to do that. So there's an enzyme called glucose 6-phosphatase, or G6-PAs. G6-PAs. That enzyme is found in the mitochondrion. Not mitochondrion. In the endoplasmic reticulum. Okay? So G6-PAs is found in the endoplasmic reticulum. That means in order to make glucose, you have to take the G6P and put it into the mitochondria, into the endoplasmic reticulum for the reaction to occur, and then you kick out the free glucose. That's actually shown in a figure here. Okay? So here's glucose 6-phosphatase. Here's the endoplasmic reticulum. Okay, so we're on the inside of the endoplasmic reticulum down here, cytoplasm site out here. What we see is the glucose 6-phosphate is brought in by a transporter, and we'll talk more about transporters next term. It's brought in by a transporter, it's converted, and then the phosphate and the glucose are both kicked back out into the cytoplasm. This reaction turns out to be very important in the liver. Turns out to be very important in the liver. In fact, it's the predominant place that you will find it. Now, because we predominantly find this in the liver, what we discover is that the process of gluconeogenesis doesn't occur in every cell. Okay? I'll show you later a figure about the primary places where gluconeogenesis occurs, but it's primarily in your liver and your kidney. That's the two most common places you find gluconeogenesis occurring. Okay. Anyway, here's the glucose 6-phosphatase. You see it's embedded in the wall, the membrane of the endoplasmic reticulum, and you see it's on the inside, so the glucose 6-phosphate has to come in order for this reaction to go forwards. Okay. All right. And that compartmentation you don't really need to worry about. Okay. So that's the process of gluconeogenesis. Now, as I said, to make a single glucose starting from pyruvate, we need 6 triphosphates and we need 2 NADH molecules. Okay? The products of glycolysis were a net of 2 triphosphates and 2 NADH molecules. So let's think about what happens if we're running both of these processes at the same time. If I have this going on in the same cell, I have glycolysis going on. Going down, I make 2 ATPs and 2 NADHs. Going up from pyruvate back to glucose, I use the 2 NADHs so they cancel out, and I require 6 triphosphates. So if I think about this as a cycle, this is coming down and going up, and coming down and going up. Coming down, I make 2 triphosphates. Going up, I use 6. Every time I go around this cycle, I lose 4 triphosphates. And what do I get? Heat. Nothing. I get basically nothing. Okay? This is totally futile for the cell. And it's for this reason that when you have a catabolic pathway and the corresponding anabolic pathway going on at the same time in the same cell, you create what's called a futile cycle. Futile cycles, in general, are not good. There are places where they're actually used, but in general, they're not good because all they do is they produce heat and they produce nothing else of any use for the cell. They cost energy. Every time you're spinning this futile cycle, you are burning 4 triphosphates. Cells have a fairly narrow margin. They don't like to waste energy if they don't have to. So futile cycles are a problem. That means, therefore, the cells go to pretty good lengths to avoid futile cycles. So the regulation that we'll be talking about in a minute between glycolysis, and gluconeogenesis is aimed at avoiding futile cycles. This involves the phenomenon I described or referred to earlier of reciprocal regulation. Reciprocal regulation occurs, and I'm going to give you a formal definition now. I mentioned what it was like the other day, but this is a real definition of reciprocal regulation. Okay? Reciprocal regulation occurs when the same molecule has opposite effects on catabolic and anabolic pathways. And I should amend that to say same molecule or same action. As I'll show you, as we'll see later, covalent modification can also be involved in reciprocal regulation. But the same molecule or the same action has opposite effects on catabolic and anabolic pathways. Okay? If we look on the screen, we see schematically the regulation of glycolysis on the left and gluconeogenesis on the right. we see some things that are clearly reciprocal regulators. The most important one is our friend F26BP. It turns on phosphofructokinase. In very, very low concentrations, it turns off fructose 1,6-bisphosphatase. This is a reciprocal regulator. It's the most important reciprocal regulator of glycolysis and gluconeogenesis because it takes very, very tiny amounts to turn PFK on and turn F26BPAs off. When we destroy F26BP in the cell, PFK loses a lot of activity and F16BPAs gains a lot of activity. So it's a reciprocal regulator, very, very important reciprocal regulator. We also see AMP is a reciprocal regulator. AMP turns on PFK. It turns off fructose 1,6-bisphosphatase. Does that make sense? Well, actually, it does make sense. When do we make AMP? Well, I'll tell you when. So when we take ATP and we're burning energy, what we're doing is we're converting ATP to ADP, right? That's not AMP. But when we get really low on energy, we start running out of ATP. So cells like to have a backup way of making ATP. So they have an enzyme called adenylate kinase, and you don't need to know the name of that. Adenylate kinase, which takes 2 molecules of ADP, and it converts it into 1 molecule of ATP and 1 molecule of AMP. So it's actually making ATP, and the byproduct of that is AMP. So cells will only have AMP when they have low energy. They've started burning up an awful lot of their ATP, and they need more. When they need more, of course, the way to get more is to go through glycolysis. So by making AMP, what the cell is doing is it's turning on PFK, and it's turning on glycolysis that is going to turn on the production of ATP. Okay? I won't talk about citrate, but you can see it's a reciprocal regulator. This will be more relevant for us when we talk next term about that. Okay? And one question is, well, how many of these do I have to memorize? Okay? I think you should know the ones that I talk about here. So citrate's not one of those. Proton is not one that I'll talk about here. We'll actually talk about that next term also. But F26BP and AMP are very important because they're reciprocal regulators. Down here, we don't see reciprocal regulators. Okay? We don't see reciprocal regulators directly, but in a sense we do. All right? Now, look at what turns off pyruvate kinase, ATP. That makes very good sense because this is a very energetic reaction. It's making a lot of ATP, okay? And so we don't want to keep it going if we have plenty of ATP. So ATP is turning off this enzyme. ATP is an indication of high energy in a cell. When you have a lot of ATP, the cell has abundant energy. On the other hand, when you have a lot of ADP, the cell has not so high energy. It has low energy. ADP turns off gluconeogenesis. That makes perfect sense. We don't want to try to be making glucose if the cell doesn't have any energy to do it. So this is an allosteric inactivator of this enzyme. Same thing as turning off pyruvate carboxylase as well. Okay? Now, I want to say one word about PEPCK because it exhibits, remember I mentioned earlier in the term that there are 3 different ways of controlling the activity of enzymes. I said one was allosteric. The second one I said was covalent modification. And the third one I said was the control of whether or not an enzyme is made. And it turns out that's the most important control for this enzyme right here, PEPCK. ADP doesn't have a very big effect on it. The bigger effect is on this enzyme right here. If most of your cells are not making this enzyme, then you can't do gluconeogenesis, is what it amounts to. And guess what cells make this enzyme? Liver and kidney cells. So your skin cells don't have this enzyme. They don't make this enzyme. And only when this enzyme is being made in the liver and kidney can you be doing gluconeogenesis, which is basically all the time, unless all you're doing is eating all the time, in which case you might not be making that enzyme. Okay? All right, so that's the regulation of these guys, and they are directed towards meeting the cell's needs. I've already earlier talked about F16BP and how it's activating this enzyme in that feed-forward activation that I talked about. So remember, when we have the aldolase reaction is an energy barrier, so we start increasing the amount of F16BP, which is a precursor of that. That comes down and turns on pyruvate kinase, which now pulls all these other reactions and makes the aldolase reaction become favorable. That's the pulling part of pushing and pulling. Yes, Laura?
Is ATP not be considered a feedback inhibition for
Can ATP be considered what?
would we consider that a feedback inhibition of pyruvate kinase?
No. So feedback inhibition occurs when an enzyme is turning off the first enzyme in a metabolic pathway. That is, the last product of a pathway is turning off the first. So that's not feedback inhibition. We'll have examples of feedback inhibition. One you've seen so far is CTP on ATCase, and that's because CTP is the end product of that pathway. ATP is not a product of any of these metabolic pathways. Yes, sir?
So did you say both CAP-CK and pyruvate carboxylase and liver, or just one?
So it turns out that they're both produced in the liver, but this one is only produced in the liver and kidney. In other words, all cells will produce pyruvate carboxylase, and they do it for other reasons, not gluconeogenesis. But if they don't have PEPCK, they can't make glucose. So PEPCK is made only in the liver and kidney. Okay. So that's the regulation. We're going to see when we talk about glycogen metabolism that all of these things here also relate to glycogen metabolism. And that gets a bit complicated, but it's also very interesting in understanding sugar metabolism in the body. Okay. You might wonder how it is that we make F26BP. You might say, oh, no, I really don't. Please don't tell me, right? Okay? But you know I'm going to tell you. And it turns out that this is made by an interesting enzyme. It's called phosphofructokinase 2. I mentioned it earlier. It's called PFK2. Okay? PFK2 catalyzes the conversion of fructose 6-phosphate into fructose 2,6-bisphosphate. Okay? Fructose 6-phosphate into fructose 2,6-bisphosphate. Okay? So here's this confusing figure that you're going to love. Okay? So here's the reaction, all right? So we can either go in the fructose 6-phosphate down to fructose 6-phosphate up to fructose 2,6-bisphosphate. That's the reaction I'm talking about. Well, I need to tell you a little bit about PFK2 because it's a really interesting enzyme. It turns out PFK2 has two components. Two components, okay? And only one component is active at any given time. Okay? PFK2, okay, is active when the enzyme is not phosphorylated. So when this complex, and these two joined by this thing, are one enzyme. There's two different activities in one enzyme. Only one activity is present. When PFK2 is active when there's no phosphate on it. On the other hand, if we put a phosphate onto it, what we do is we inactivate the PFK2 part and we activate the other part. And what the other part does is it breaks down fructose 2,6-bisphosphate back into fructose 6-phosphate. So you put a phosphate on it, the phosphatase is active, that's called FBPase 2. If we take the phosphate off, the PFK2 is active and it makes F26BP. Now, what puts the phosphate on? What puts the phosphate on? There's our friend protein kinase again. When is protein kinase going to be active? Under what kind of stimulation? What hormone? Epinephrine. So when epinephrine is present, protein kinase A is going to be active. We are going to favor the production of the phosphatase. We are going to favor the breakdown of fructose 2,6-bisphosphate. And when we do that, do we favor gluconeogenesis or glycolysis? We favor gluconeogenesis. Okay? Now, you think, oh, that's confusing. Aren't I breaking, doesn't epinephrine doing something so that I have more energy? Right? Isn't that the way you thought about that? Okay? It turns out that epinephrine is not there for energy directly. It's there for glucose. The reason that you get the energy is your liver starts dumping out glucose by the tons. That happens by the breakdown of glycogen that we'll talk about next week, and by the synthesis of glucose in gluconeogenesis. Both of those are aimed at increasing the blood glucose levels. When the blood glucose levels go high, the muscles have a lot of energy, and you can run and you can escape. So in epinephrine binding, we go to the right, and we favor gluconeogenesis. If we bind insulin, on the other hand, we activate the phosphoprotein phosphatase, which takes off that phosphate. Now we start making fructose 2,6-bisphosphate, and we activate PFK and we activate glycolysis. Activate gluconeogenesis by epinephrine, activate glycolysis by insulin. Why are we doing glycolysis with insulin, you're thinking? Don't we already have plenty of sugar? Right? Yeah, we have plenty of sugar. The cell has taken up a lot of sugar, and what is glucose? It's a poison. And what do you do with a poison? You get rid of it. You take some of it, you make it into glycogen, you take the rest of it, and you break it down. And that's what's happening. Okay, now, this figure I will tell you most of you will find confusing, and I could spend another 1,000 or so words on this, and you'll still find it confusing. The way that this is going to make sense to you is if you take it home and write it down and think of the different scenarios that I just described to you. If you can think your way through those scenarios, you understand this pathway. Okay? If you don't understand those scenarios, come see me, and I will explain. I will use my 1,000 words with you to help you understand them. Okay? Questions about that before I go? Notice it says glucagon stimulates PKA. Glucagon acts like epinephrine does. Okay. Okay. Well, I've got one more thing I'm going to talk about, and then we'll actually have a little fun. And the one more thing to talk about is the Cori cycle. And this is a cycle that I think almost all students find very easy, and it's very cool, and it tells you something about how your body works. Okay? The Cori cycle, I'm going to sort of paint a mental picture in your head before I show it to you. The Cori cycle is a sort of a communication between your liver and your muscle cells. Your liver and your muscle cells. Let's imagine that I'm doing that jog I was doing this morning, where I ran a little bit too far. I usually run about 5K, and I did about a 7K this morning, which on a weekday morning is a little too much for me. Okay? And so by the end of my run this morning, I am really huffing and puffing. So if I'm huffing and puffing pretty bad, what does that tell you? Am I aerobic or anaerobic? I'm going anaerobic. That's why I'm trying to get that more oxygen in. So my muscle cells, what are they producing if they're not getting enough oxygen? They're producing lactate, right? And they're producing lactate because they've got to keep what going? Glycolysis. Very good. Okay, you guys, this is good. I'm very happy to hear this. You guys have this down well. Trying to keep glycolysis going because they need to make more NAD. And so pyruvate to lactate converts NADH back to NAD, and I'm set. But I just told you earlier today that lactate is a biological dead end. And when did I say we made pyruvate back in, lactate back into pyruvate? When the oxygen concentration goes up. But I'm still running. I'm still running. Okay? Meanwhile, what's happening? If I don't do anything, my lactate concentrations are going to increase. Okay? My lactate concentrations are going to increase. It's not going to do my muscle cells any good. It's going to change the pH of my muscle cells because it's an acid. And I want to be careful with that. Okay? So the muscle cells, get out of here. Don't want it. The muscle cell kicks it out and into the bloodstream. That's what it does with it. Because if it stays around, it's going to change the pH, and the pH is going to have some serious effects on the cell. It kicks it out into the bloodstream, and it goes in the bloodstream and it travels to the liver. And it gets to the liver, And the liver grabs that lactate. Why in the world does the liver grab the lactate? Well, it turns out that the liver grabs the lactate because the liver has plenty of oxygen. The liver is very close to my heart and my lungs. It's not lifting weight. Okay? It's not doing what my muscles are doing, and so it's got plenty of oxygen. It takes that lactate, and because it's got plenty of oxygen, it converts it back to pyruvate. And because my liver cells have PEPCK, it's going to take that pyruvate, and it's going to make it back into glucose. Right? I'm making a little glucagon. I'm making a little epinephrine. My body is saying, hey, I need some glucose. The liver takes that glucose and says, okay, I've taken this lactate, I've made glucose out of it, I don't need the glucose, and it dumps it out into the bloodstream. And guess where the glucose goes? To the muscle cells. Now you know how the liver feeds your muscle cells when you're low on energy. This is known as the Cori cycle. It was discovered by a husband and wife team who later won the Nobel Prize, and this is what it looks like. In the liver, lactate goes to glucose. Glucose gets dumped into the bloodstream. Muscle cells take it up, they convert it to lactate, and dump it back. So it's a perfect cycle. Now you might say, well, that's a futile cycle. Does this look like a futile cycle to you? Is this a futile cycle? I say yes and no. It's not a futile cycle. What was the definition of a futile cycle? Has to be in the same cell. This is a perfect example of where organ specialization is necessary for the different needs of an organism. The liver is, we're going to see your favorite, your most favorite organ of all time because it's involved in everything. It's involved in controlling levels of glucose. It's involved in controlling levels of fat. It's involved in controlling levels of fatty acids. It's involved in control of synthesis of fatty acids. All kinds of stuff. Very important for that. Lori? Yep. You get hot because you're running glycolysis because of the pyruvate kinase reaction, or among others, but among that one. Okay? Well, I promised you fun. I have some fun. And this is a kind of a long song. But you'll notice last time when you sang along and we were loud that the average went up. So I want to hear that here. This is a song to the tune of supercalifragilisticexpialidocious, and it's called Gluconeogenesis. Ready? When cells have lots of ATP and NADH2, they strive to store this energy as sugar, yes they do. Inside a mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose, come to be. Glucose, glucose, come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerates, exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with electrons gives us all an NAD. NG3Ps isomerized to make DHAP. Oh, gluconeogenesis is anabolic bliss. Can't hear you. Reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose, glucose factory. Glucose, glucose factory. The aldolase reaction puts together pieces so. A fructose molecule is made with 2 phosphates in tow. And one of these gets cleaved off by a fructose phosphatase. Unless F626PPs acting, blocking pathways. Oh, gluconeogenesis, a pathway to revere that makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose, glucose, jubilee, glucose, glucose, jubilee. From F6P to G6P, this is the final phase. The enzyme catalyzing it is an isomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard, because our professor lets us use a filled out index card. Gluconeogenesis is not so very hard. Okay. Whew. Thank you. I'm out of breath. Happy Turkey Day. Okay, how you doing, dude?
That was a long one.
That was a long one.
Practically, they were like, can you have
Yes, sir.
moment?
Yeah, what's up?
I missed,
Uh-huh. Oh, that wasn't me, I guess. Okay, yeah, what's up?
sorry if I missed this in the beginning. I jumped a couple minutes.
Uh-huh.
But I know we normally have 2 days to submit.
Yeah, you'll have until next week. Don't worry, that's fine.
Oh, okay.
Yeah.
When would that be then, since we have a holiday?
Well, a total of 3 days, 3 school days.
Oh, okay.
Yeah,
3
yeah.
school days? Okay. And
Okay.
the key is posted outside your office?
Yes, it is.
All right, perfect. Thank you.
Thank you.
Yes.
I found the building. I ran out really